Welcome to Daily Papers, the podcast where we break down the latest and greatest research into bite-sized pieces. Today, we're diving into the microscopic world of our own bodies and a new way to make sense of it all.
Imagine trying to understand a massive city, not by looking at a map, but by trying to read a list of every single phone call, text message, and conversation happening between millions of people all at once. It would be an impossibly tangled mess.
That's pretty much the challenge biologists face every day. They work with what are called biological networks, these incredibly complex webs of interactions between all the genes, proteins, and cells in our bodies. Understanding this web is the key to understanding health and disease.
And these networks are getting unimaginably large. We're talking about data with hundreds of thousands of components and sometimes billions of connections between them. It's a classic big data problem.
For years, scientists have been developing artificial intelligence to try and untangle these webs. The current state-of-the-art tools are called graph neural networks, or GNNs. They're designed specifically to work with network data.
But they have some serious limitations. When a network gets too big and dense, GNNs can get overwhelmed. They're computationally expensive, meaning they need huge amounts of processing power. They can also over-smooth the data, kind of blurring all the details together until everything looks the same.
And maybe the biggest problem is that they are often a black box. The AI might give you an answer, say, this cell looks cancerous, but it can't really tell you why it thinks that. For scientists and doctors, the why is everything.
Exactly. Which brings us to today's paper from researchers at Stanford University. It's titled Transformation of Biological Networks into Images via Semantic Cartography for Visual Interpretation and Scalable Deep Analysis.
That's a mouthful, but the core idea is brilliant and actually quite simple. What if, instead of trying to teach an AI to read that impossibly tangled web, you first turn the web into a picture?
That's the whole idea behind their new method, which they call graph-to-image. It's a clever process of biological map-making, or as they call it, semantic cartography.
Here's how it works. First, the algorithm looks at the giant network and identifies closely knit communities of nodes. Think of it like finding distinct neighborhoods in that giant city of conversations.
Then it uses a really clever mathematical technique called optimal transport to arrange these neighborhoods on a 2D grid like a map. And the key rule is this: neighborhoods that interact a lot in the original network get placed close to each other on the map.
This creates what they call a structural image, a picture that represents the structure of the network. They do a similar thing for the features of each node, like its gene activity, creating a feature image. Then, for every single node in that original giant network, they create a unique multi-channel image by layering these maps on top of each other.
It's like creating a custom satellite image for every person in the city. One layer shows their social connections, another shows their daily activities. And once you have an image, you can bring in the real heavy hitters of AI.
You're talking about convolutional neural networks, or CNNs. These are the AIs that are famously good at image recognition. They power everything from self-driving cars to facial recognition. We have decades of research making them incredibly powerful and efficient at spotting patterns in pictures.
So the big question is, did it work? Is turning a biological network into a picture actually better?
Oh, it worked. The results are pretty staggering. In one test, they gave the model a network of human proteins and asked it to classify which of 54 different body tissues a gene was most active in.
So is this a gene for your heart, your brain, your liver, that kind of thing?
Exactly. The best of the old methods, the GNNs, got it right about 45% of the time. Not bad, but not great. Graph-to-image, however, had an accuracy of over 83%. That is a massive leap in performance.
And because it's based on images, the researchers could peek inside the AI's brain to see how it was making decisions. They found that it wasn't just guessing. It had learned to group tissues in ways that make perfect biological sense. It automatically rediscovered entire organ systems just by looking at the data.
Then they decided to really push the limits. They used a dataset of mouse cells that created a network with over 54,000 nodes and over 3 billion connections.
3 billion, that's enormous.
It's so big that the standard GNNs couldn't even run on the full dataset. The researchers had to give it a simplified version that was 99% smaller, but graph-to-image handled the entire massive network on a regular personal computer, and it was still more accurate.
That's incredible. It's like the new method isn't just better, it's playing a completely different sport.
My favorite example was the clinical one. They used it on a prostate cancer dataset. The task was to tell the difference between a primary localized tumor and a much more dangerous metastatic tumor that has started to spread.
A hugely important distinction for treating patients.
Graph-to-image could do it with 99% accuracy. And again, it wasn't a black box. The researchers could see exactly which biological signals the AI was looking at. It had learned to identify the molecular hallmarks of aggressive cancer, things like increased metabolism and protein production that human pathologists are trained to look for.
It acted like a molecular pathologist in a computer. So, to sum it up, this paper introduces a completely new way to look at biological data. By turning these abstract, complex networks into concrete images, we can analyze them with AI tools that are not only more powerful and more scalable, but also more interpretable.
It really is a paradigm shift. It takes a problem that required specialized, complex AI and transforms it into an image recognition task, which is something we're already fantastic at solving.
This could open up new doors for everything from diagnosing diseases earlier to discovering new drugs, all by helping us finally get a clear picture of the tangled web of life.